Very good afternoon everyone. Now we are going to study about LAC OPERON, which was given by Jacob and Manu. So when you read this topic in NCERT, you might feel a little bit confusing. But if you understand this topic, you won't find as easy as topic than this. You won't find a topic which is easier than this. Trust me, this is one of the most easiest topic in molecular basis of inheritance. Let's look into it. So let's imagine something. You have a mango shop. So you have an estimation that every day you get around 150 customers to your shop. What will you do? Will you make 150 liters of mango juice early in the morning, or you will make the mango juice whenever the customer comes to you? What will you do? Will you make the mango juice early in the morning for all the customers, or will you make the mango juice whenever it's needed? You will choose option 2. Whenever it's needed, we will make the mango juice. It's the same thing happens in E. coli. So what happens is, so there is a E. coli bacteria. So now in its surroundings it has glucose, and it obtains and gets energy. So in its surroundings it has glucose, it has taken the glucose and it got energy. Sometimes glucose is not present, sometimes lactose is present. Now what this E. coli will do is, this lactose cannot be directly taken into the E. coli and cannot get the energy. It needs to be broken down. So in order to break it down, break the lactose down into two parts, it needs some enzymes. So E. coli releases some enzymes, only then lactose will break into glucose and galactose. So sometimes glucose is present. So when glucose is present, it can easily take it and it can get the energy. But when glucose is absent, sometimes lactose is present. Lactose cannot directly convert it into glucose. So it needs some enzyme to break the lactose. Only then it can get the energy. So now imagine, will E. coli produce the enzyme all the time? So if it produces the enzyme all the time, it's waste of energy for it. Because sometimes when glucose is present, there is no use of these enzymes. Right? When glucose is present, there is no need of using these enzymes. So this energy conserving method is known as LAC OPERON system. So they will release these enzymes whenever lactose is only present. When glucose is present, they don't release these enzymes. They will release enzymes when lactose is present. So this is the concept of LAC OPERON. I hope you understood it. So link this with our mango juice shop. So we will produce mango juice. We will make the mango juice whenever it's needed. Whenever it's needed. In the same way, E. coli will produce the enzymes whenever it's needed. Remember this term? It says everything. That's it. And coming to this, it is present in E. coli and it regulates the lactose metabolism, and it is inducible operon. It has on and off system in it, and it is an energy conservation system, because if they release the enzyme all the time, it's a waste of energy for them. That's the reason why these are energy conserving method. And coming to components of it, we have a repressor protein, LAC-I, and second one, RNA polymerase binding site called P, promoter region, and repressor protein binding site called O, operating region. And we have structural genes, LAC-Z, LAC-Y, LAC-A. LAC-Z, it produces an enzyme called galactosidase, and LAC-Y produces permease, and LAC-A produces transacetylase. So these enzymes are same. So when lactose is present, these enzymes are released. So this is the enzyme which breaks the lactose into glucose and galactose. Remember, this is the enzyme which breaks the lactose into this enzyme permeates. This is like an indicator. When there is a lactose outside the E. coli, it will indicate, it will tell the E. coli that lactose is present outside. So then lactose will enter into it and it will break down with the help of LAC galactosidase. And coming to LAC, which releases trans-acetylase, there is no much information about it. We don't know what's the function of it. So remember till this, and we will draw the diagram now so that we can understand it easily. First, let's draw the components. Regulatory chain, where the first one, repressor protein is present. Repressor protein, call it as the chain is LAC-I. And here we have P, means RNA polymerase binding site, promoter region. Here we have O, repressor protein binding site, operator region. And here we have structural genes, Z, Y and A. Right? Now there is no lactose. No lactose. So remember this diagram. Here we have regulatory gene where repressor protein is present, LAC-I. Here the RNA polymerase will bind here, promoter region and O. This is the region where repressor protein binds, and here we have structural genes. So now there is no lactose is present. What will happen? So this repressor protein will bind with the operating region. So RNA polymerase comes and binds with the promoter region. Now the repressor protein is binded with the operating region. So this RNA polymerase cannot start the transcription. It will stop here because the structure of operating region has changed. It cannot let the RNA polymerase to move forward. As it cannot move forward, they cannot be transcribed. And now we don't get these enzymes. Understand? There is no lactose is present outside. So here the repressor protein binds with the operating region. So from outside we get RNA polymerase which binds with the promoter region, and this binding changes the shape of the operating site, operator region. So it won't let the RNA polymerase to move forward. It cannot release or it cannot transcribe the structural genes. As a result, no enzymes are produced. No enzymes are produced. Now, let's see what happens when lactose is present. So I hope you understood. This repressor protein binds with the operating region so that RNA polymerase cannot move forward and it cannot transcribe these elements. And those enzymes cannot be released if the transcription doesn't happen. So this is what happens when there is no lactose. Let's see when there is lactose is present. Lactose is present. Previously we were talking about off system. When OPERON, LAC OPERON is off, means lactose is not present. Now we are talking about on. LAC OPERON is an on state. So now we have lactose. So this is converted into lactose, which is an inducer. So this binds with the repressor protein. So now it binds with the repressor protein. Normally what happens here when LAC OPERON is absent, when lactose is absent, the repressor protein goes and binds here. When lactose is binded with the repressor protein, the structure of repressor protein will change. It will change the entire structure of repressor protein. When it tries to bind with the operating region, it cannot bind as the structure is different. It's like a lock and key. So, can you open your room with other room keys? You cannot open. In the same way, the structure of repressor protein is changed when lactose binds with the repressor protein, so that it cannot bind with the operating region. So now, when RNA polymerase comes and binds with the promoting region, it goes through the operating region and structural genes, and the transcription happens, and we get galactosidase and permease and transacetylase. So this is what happens when lactose is present outside the E. coli. So when lactose is present outside the E. coli, it releases galactosidase, permease and transacetylase. So these enzymes cannot be released when lactose is absent outside. So there is no need to release those enzymes when lactose is absent, because these enzymes helps lactose in breaking down, and it helps in making lactose enter into the E. coli cell, and we don't know the function of transacetylase. So now let's revise what happens here. So now the cells have recognized that the E. coli has recognized that there is a lactose present outside the E. coli. So there is a little amount of permease is present in the E. coli. It lets the lactose inside, and it is converted into lactose, which is an inducer. It binds with the repressor protein, and it changes the shape of the repressor protein so that it cannot bind with the operating region. So now it cannot bind with the operating region. Now RNA polymerase comes and binds with the promoter region. It travels through the operator region and transcribes structural genes. And this LAC-Z, LAC-Y and LAC-K will release the enzymes galactosidase, and Y will release permease, and A will release transacetylase. So this galactosidase will help in lactose breaking down into galactose and glucose, and then the E. coli will get energy. So this is how E. coli regulate the lactose metabolism. So they release enzymes whenever it's needed. That's it. So this is about LAC OPERON. I hope you understood it. I tried to explain it in a very easier way. Thank you for listening. See you in the next video.